Dr. Rossi, thank you very much for spending some time with us today.
Oh, my pleasure.
So I wanted to ask my first few questions on career development. The new investigator committee of ISEH aims to promote the career development of trainees in the field of hematology. So I was wondering if you could talk to us about some key events in your training that helped you develop your successful career.
Sure. Well, I actually took a very non-linear path, a very atraditional path through my career in science, which is not what most people do. So I actually started a graduate program, a PhD program in Toronto, which I didn't complete because I moved to Paris, France. I was in yet another PhD program there, which I didn't complete because I got exhausted. And then I moved to Texas. And so my third PhD program was in Helsinki, and that one I actually got. And along the way, though, I did a lot of science. I worked on early embryonic development to gastrointestinal polyposis to peripheral myelination. So I got to view this really broad swath of biology and molecular biology. And so, though I ended up sort of doing, my postdoc and getting my faculty position, later in life compared to most people, because some people would argue, well, I took all these strange detours, but I would actually argue that that was really informative for, giving me a real big perspective on biology, and which I wouldn't trade for anything, actually. I really feel like that was really important for me when I eventually became a full-time scientist and not just bouncing around the planet. I really had a deep expertise, such that even by the time I did my PhD, I was, publishing many good papers, and as a postdoc as well, because I had done science for so long and had, was quite good at it. So that's an unusual career path that most don't take. Although I would, and I've mentored people to take things slowly if they possibly can. There's plenty of time to be an adult later on in life, but being a young person and getting as much exposure and becoming a rounded individual is I think really important for one's later career as an adult. Can give you some creativity and some perspective that those that follow the real fast track don't have. I remember, for example, I was in my office at Harvard Medical School, and this new freshman, Harvard freshman, came into my office, and he was 17 years old. This skinny kid, he couldn't have been more than 85 pounds. And he had sort of advanced through all his schooling so early. And even before he started this was in the summer before he started his freshman year, and he came into my lab wanting to be in the lab and start to work. And I said, well, don't you think it's a good idea to pause for a moment and go into your undergrad and see how that is, and start to decide that you have the bandwidth or what you're interested in. So I tried to advise this kid, this very young kid, to take time, get experience, because I think it's actually really beneficial. So like I said, it's atypical, but it served me well, that's for sure.
Can you describe to us some of the key mentors that motivated you during your training?
Sure. Well, there was many, and this is another great thing about this path that I took, is that I worked with really smart people around the planet. So, dating back to my original graduate program in Toronto, Alan Bernstein, who I'm in contact with now about sort of Canada's response to the COVID pandemic, for example. Then I worked with Gilles Thomas and Olivier Delattre in Paris, and they were geneticists that were taking their genetic findings and turning them into sort of biochemistry and molecular biology, and that was interesting. Then I worked with Tomi Mäkelä in Helsinki, and he had just come out of Bob Weinberg's lab. He had sort of just started his own lab in Helsinki. So he was a real rising young star, smart guy. And he had trained with Kari Alatalo in Helsinki. He was a great scientist. And then I ended up in, when I went to go do my postdoc, I had by this point published so many papers that I literally could have gone to any lab I wanted to. I applied to 11 and I got offers from all of them. And these were big, big labs on the planet. But I decided to go work with Irv Weissman because I wanted to focus on stem cell biology and the intersection with aging. And, Irv, of course, is well known to the ISEH community. He's brilliant. And he was a great mentor to me. And then even though when you start your faculty career, you still need mentors. So when I got to Harvard, Fred Alt, who was my department chair, a great scientist, is a great friend and mentor to me, as was Doug Melton, who was chair of my department. So you always need mentors, no matter what stage of your career you're at, and they can really help you think about getting it right, quite frankly.
Got it. And in retrospect, what kind of skills did you acquire that you think were the most useful to navigating your career as a scientist? Or
yeah, well, well, the one thing I would say is when you have a new scientific, a direction you want to go in, sometimes you have to invent the wheel if the wheel isn't already in place. Let's say you come up with your science idea and the only wheel available is a square wheel, in which case you might have to innovate and come up with a round wheel. On the other hand, I would argue that if there's already somebody in your community who's perfected the round wheel, you should collaborate with that person, reach out and be really open about your science and get experts involved. So that's how I approach things. Even as a postdoc, I remember approaching my fellow Irv Weissman had a huge lab. There was 40 people in it, and when I had something that I wanted to do, and there was somebody else in the community that was really good at it already, I would immediately reach out to them and start to collaborate with them. So, I guess the message is, often in the settings that we're at these universities and teaching hospitals around the planet, there's some really great people that are already experts in what, some aspect of what you want to do. So you should always be open with your science and reach out for collaboration, because that's the best way to move science forward.
Okay, awesome. And can you describe to us your first contact with ISEH and how it played the role in your career?
Well, I don't know about my first necessarily, but it's at least the annual meeting was my favorite meeting every year, because it's really focused on hematology and stem cell, hematopoietic stem cell biology in particular. So it's actually a meeting that I really enjoyed going to every year. It's the one that was really focused on what I was interested in regards to hematopoietic stem cell biology. I think at some point ISEH offered me an opportunity to speak at the annual talk as a new young investigator. It was a great honor, and went to the meeting and gave a talk in this particular session. But yeah, like I said, that's as opposed to and I've been a member of ISSER for forever, and that's a great meeting too. But of course, it's not just blood focused. That's great too, because sometimes it's really important to see, get outside of your box. That's actually a really important, lesson. Get outside of your box and learn some ideas that are in totally different fields, and then be inspired to apply them to your work. For example, Yamanaka's work, which I first saw at ISSER in, I think it was 2005, just prior to his publication in 2006, that talk, so to say, blew me out of my chair in the conference hall. It was so fantastic. I loved the science. And then we went on and we're inspired by that in my lab. And we did a number of projects first, using a multi-transcription factor approach to trying to reprogram blood cells back to hematopoietic stem cells, but also our efforts in, generating integration-free iPS cells led to this, modified mRNA technology that, now people are getting in their arms.
Mm-hmm. Do you have any advice for people who are starting their own lab in regards to what kind of projects they should take on at first? Or
Yeah, well, again, this is probably not good advice. This is what I did. So I did not. Often you write your grants, and you write your, or even when you're starting your lab, you've got a sort of proposal that you've laid out your first 5 years or 3 or 4 years or whatever. And some people want to stick to that plan. I think that's a terrible idea and I also think it's a terrible idea to write a 5-year grant because, of course, the discoveries that you'll make in year 1 and 2 will inform where you go in years 3, 4, and 5, and so on. Always the discoveries you make inform where you go. So I think a long-term plan is quite silly, actually, because it makes no sense scientifically, because, like I said, you haven't seen the early data yet, so how can you know where you're going to go? So my advice is to really go where your interest lies and go where you're passionate, and don't go where your grants said that you were supposed to go. Mm-hmm. So I know there's a bunch of granting agencies out there that want to reach through the webinar and grab me by the throat and strangle me right now, because that's not how it's supposed to be. You're supposed to do what you said you were going to do, but I never did that. I always, I had a pool of money from many different grants and funding agencies. And then we just followed whatever science was interesting and whatever, wherever the data led us. And honestly, you write the grants. Like I said, I feel like it's a really a silly exercise for 5 years in advance. I don't understand that. It makes no sense at all to me. But you have to do it, unfortunately. But I don't think you should stick by that roadmap. You should, where the data and the passion leads you is where you should go.
And in regards to that very successful translation of discoveries from the academic lab into startup companies and then to the clinic, did you already have this plan in mind when you set out to start your own lab, or it came as things progressed?
Yeah, no, I didn't. I was pretty happy and content and had, like many young people, a career in academia as my focus. And it's a great job. It's, one of the best jobs you can imagine. And though I had seen translation from the lab to biotech from my time in Irv's lab, because he had translated several ideas out of his lab to biotech startups that ended up in the clinic. And sometimes he didn't even do it via, or Irv's an amazing person, sometimes not even via the biotech route, but rather just sort of kept it in the lab and then ran investigator-led clinical trials from his lab and from collaborators' labs. So I was interested in that, and got my first real good look at it when I was at Stanford during my postdoc, and I was working on aging and hematopoietic stem cell aging, and I thought, there might be an opportunity if we made a great discovery to move something, but in all the time that we even worked on it in my lab, not once did I start an aging company, because really there wasn't a tractable idea there. So the first real experience I had of doing it myself was with modified RNA, and the work that we were doing, as I just mentioned, is inspired by Yamanaka's work, where we were trying to make integration-free DNA, integration-free iPS cells. At the time, that was very important, still very important for moving those cells to the clinic. There was a lot of different labs working on different strategies. Project led by a postdoc in the lab at the time, Luigi Warren, had this idea to use mRNA to generate integration-free iPS cells. And there was a lot of work that we did over years that then led to this modified mRNA concept, which was really required to do this. And then it was funny. I was, as we were getting ready to publish our paper, word was getting out because it was an exciting story at the time.
Yeah.
And so all of, these pharma reps were calling me. I'd get a different call each week from some sort of pharma rep saying, hey, we'd really like to license that IPS technology from you. We really want to get, because all pharma was, trying to use IPS disease modeling in their labs at the time, which is a great idea, and it's really panned out. Mm-hmm. But what I was surprised by was that nobody in any of those calls recognized that the modified mRNA technology itself, the ability to express any protein that one wanted, to me, that was the important thing. And if you could translate that in vivo, now all of a sudden you had an ability to do gene therapy without the genes, without the DNA. And, we demonstrated that, you could use this technology temporarily, express something whenever you wanted. It was dose dependent. You could repeat dose if you needed to. So many properties of it made it really exciting for a possible therapeutic translation. So that was, that's what I founded Moderna on, was the idea of translating this technology towards therapy. It's funny because, Moderna was in stealth mode for many years and people just thought we were working on iPS cells or something like that. But there was not a single experiment ever done in Moderna on iPS cells. It never had anything to do with that. It was all about turning this modified mRNA into a therapeutic itself, which, now 10 years later has borne fruit with this.
Wow.
Like you mentioned at the outset, this COVID vaccine, for example.
That was awesome, and such a fundamental discovery about biology and how it can be translated to the clinic. And so did you have an idea of what disease application as you founded Moderna, or already in mind, or?
Well, not a particular one, because part of the challenge with such a technology, it was an abundance of riches. You could work on so many different things. Human disease is, almost, invariably comes back to some sort of misexpressed protein or misfolded protein or some aspect of protein biology. If there's, any genetic disease, for example, there's a mutation in the gene, leads to a bad mRNA, leads to a bad protein, 6,000 genetic diseases. And I was trained in molecular genetics, so I was thinking about genetic disease. On the other hand, you had to be thinking, okay, there's a new technology. mRNA is a macromolecule. It's a big molecule. So you had to think about getting it into, efficient delivery. The good news for us is that, small interfering RNA technology had been developed sort of 15 years prior, and they had already gone through the challenge of delivering even, much smaller, siRNAs, but still the concept was there. So via lipid nanoparticles, for example, turns out we did experiments in the lab. You could even put in unprotected mRNA, not in a vehicle, and get expression. It was pretty amazing. So really it was, what do we work on? And I will say that vaccines were not high on the list at the time. And although it's a great application for the technology, it's a perfect application, because you have to give one dose to a patient who's never seen an antigen before, and you can non-invasively measure their peripheral blood to see if they've responded to it and to see whether or not it's been safe or effective. So you can do it intramuscularly. So it's a very good application for it. And indeed, that's how Moderna ended up getting into it. Already some years ago, well before COVID. But so I was thinking genetic disease primarily. We did a couple of proof of concept studies in my lab that were actually never published, but it was really just to see, could we take this mRNA therapeutic, this modified mRNA, and have some sort of therapeutic outcomes. So I remember some experiments that were done by a postdoc in my lab, Pankaj Mandal, and Wataru Bina was a grad student, and Morag Stewart, where we made a human erythropoietin, which is, there's a ELISA assay that distinguishes between mouse and human. So we made human, we injected it into the thigh muscle of the mouse. And again, you could measure the peripheral blood to see whether or not we were making human EPO, which we were, again, in a dose-dependent manner. And then, of course, you could measure the red blood cell production in the mice, which we did, 14 days later. And sure enough, their red cell counts went up in a dose-dependent manner and hematocrit. So it told us that we could put a human gene, basically an modified mRNA for a human protein into and by the way, this worked on the first time we ever did it. And that tells you about something about the robustness of the technology as well, when you can take something and get it to work right off the bat so nicely. So we did that experiment. We never published it. We did that in 2010 or early 2011. We also, made luciferase because, that was the first time we went, actually the first time we went in vivo was GFP, but luciferase is a good way to look. And again, the first time we did it, we did an intramuscular injection into the thigh muscle of the mice. And again, in a dose-dependent manner. These were experiments done by Lee Orazangi and Pankaj Mandal in the lab. And we, again, saw dose-dependent luciferase, expression in the thigh muscle of the mice, again, unpublished. Then we also went into the heart, we went into the lung, we started exploring different tissues with various collaborators, and every time we went into a different tissue, we get expression of modified RNA. And this gave us really...
Just imagine the excitement of these results.
Yeah, totally. And it of course, I had just founded Moderna at the time, and these were the key experiments that said, well, because prior to that, the study we did that was published in Cell Stem Cell, that was all ex vivo, it was all in vitro, transfecting cells, and it was a very nice study. I mean, we expressed 7 proteins at the same time, and reprogrammed cells and directed their differentiation. Really, really a lovely study, but all cells in a dish. Mm-hmm. So could have been that it, took us, maybe we never succeeded in getting it in vivo, but it turns out, like I said, there was enough done prior to getting nucleic acids into animals. Despite being such a gigantic macromolecule, it is possible. And you only have to go through one membrane. You just have to get through the cell membrane. You don't have to get into the past the nuclear membrane, because the ribosome lives in the cytoplasm, and that helps. So yeah, it was pretty exciting.
That's very cool. And in terms of navigating all of the steps from protecting the technology to licensing it and discussing with investors, taking it from an institution to a company, how did you learn to navigate all of these various steps of
on the fly? Of course, we're not trained in how to do that at all. And you can read books and you can talk to people about how they do it. But ultimately, the good news was that the science was spectacular. So the science really spoke for itself, which really helps. And, I was very good at giving a science talk. Obviously, it was work that came out of the lab. So you could really captivate people's attention. So really the first investor, for example, that we got, it's interesting, was, so I was at it was then called the Immune Disease Institute. It's now called the Program in Cellular and Molecular Medicine at Boston Children's Hospital. But we had sort of a monthly faculty meeting where just the faculty would, one person would present their data. And I presented this study at the faculty meeting to, my faculty, or the people in our, maybe 13 or 14 faculty members, and they were very excited by it. But there was one person who was unable to make it then. It was a guy named Tim Springer, who's a great scientist. And Tim had great success launching a company himself years previously, a company called Leukocyte, which was then sold to Millennium. He had wasn't there at this day. So somebody made the suggestion, oh, Derrick, you should really go tell Tim about these studies and get his thoughts on this. So I said, yeah, sure, happy to. And I was pretty junior faculty at the time. This was 3 years into starting my faculty position. So I went over to Tim's office and then showed him the data. And I remember it was a really odd meeting because he was being really aggressive with his questioning, like in a way that was, I found a little bit weird because, you know, here's just a faculty showing another and he was really just peppering me really hard with questions about, you know, IP and where's it going and just all kinds of things. Thought it was a little strange. Anyhow, at the end of going through this sort of, you know, strange 45 minutes or an hour and a half, can't remember, you know, Tim's visage totally changed. And, you know, this big smile, and he said, Derrick, this is fantastic. I want to invest in this. So Tim, you know, at that moment became the first investor in Moderna. He didn't write me a check in his office, obviously, but he also helped me because he had done this before. So he put me in touch with, you know, some VCs. I had already reached out to some VCs, venture capitalists myself, but Tim had other connections. And he also put me in touch with Bob Langer, who's this prolific inventor at MIT who had started many companies. And Tim and I made the trek over to Cambridge side, you know, Harvard Med School is in Boston. So we went across the river, the Charles, met with Bob Langer. And, you know, again, I took Bob through the whole science deck. And then, you know, Bob's a very smart guy, obviously. And, you know, at the end of it, he said, this is terrific. What can I do to help? And, you know, I thought, well, what can he do to help? As we were traveling back, you know, Tim and I back over to Boston from Cambridge, I thought, what can he do to help? And I thought, well, maybe he could help me co-found this company because he's done it many times and I have not. And he's a delivery expert. That's his expertise. And ultimately, we've got a macromolecule that we're going to have to deliver. So then I reached out to Bob. Maybe we had one more meeting, but, you know, maybe after the next meeting, I said, Bob, would you be interested in co-founding this company with me? And so it really helped to have a very experienced biotech entrepreneur, you know, help me navigate the rest of the process for starting the company up. So, you know, it was fortuitous. It's connections, it's your colleagues, it's the people around you that have experienced, those mentors, really critical. And you can't be alone, and to do it your first time would be very challenging if you were not doing it with somebody that had expertise and you know, connections are important, obviously. So it turned out to be a nice story, obviously.
Yeah, that's very nice. And at the level of the institution itself, in terms of protecting did you have to overcome any hurdles in taking the discovery from an academic lab to a private?
No, well, we had a really small tech transfer office at it was then the Immune Disease Institute. It was a small Harvard affiliate. So we had a small tech transfer office. We did do a very, very broad, big IP, the initial IP. So to try to get as much protection as possible. If I had to do it again, I might do it a little bit differently because then I, you know, sort of learned a lot more about IP. This was my first major piece of IP that we were filing, intellectual property. But the good thing is that the Institute was supportive of, you know, me launching a company and the Institute licensing that IP to the company. Whereas, you know, sometimes your institute might not do that. They might, you know, well, we got a better offer. We're going to make, you know, some X amount of dollars more if we license it off to, for example, pharma was knocking on the door and they wanted to license it for the iPS cell aspect of it, which, you know, turned out, which is a great application, but it's not the real, you know, the real value of the technology is more in the therapeutic domain. Clearly, So the Institute was supportive of whatever I wanted to do with it. They would be supportive, which is odd. Maybe not odd, but I was a very junior faculty. I mean, I was 3 years into my faculty position, and I could have made a mess of it, but luckily I didn't.
And generally, do you think that academic researchers should be more active in translating their own research results?
Well, I don't know about more active. I mean, the fact of the matter is that some discoveries are translatable and they have a path to development for clinical application, but 99% don't. Mm-hmm. We just have to be clear about that. And so young people are always sort of pitching ideas to me. This seems to be what I spend a lot of my time doing, people reaching out saying, hey, we got some really great science. Would you like to take a look at it? And I always do, and I'm happy to. And sometimes the science is fantastic, but how on earth are you going to take that forward from the perspective of raising money and getting into the clinic and developing a clinical trial that could really tell you something? So though sometimes discoveries come in very out of left field, like CRISPR is a good example. A bacterial immune system against bacteriophage. Right. That's basically what CRISPR is. And though when it made the jump to human cells for targeted gene editing, it was very clear that okay, now you can do something with that. And indeed, my lab was very involved in the early days. As soon as CRISPR jumped into mammalian cells, we were, I think, the first lab to take it into hematopoietic stem cells, and we published a paper on it in Cell Stem Cell targeting CCR5, work of Pankaj Mandal and others in the lab. So I think you have to have a little bit of a sense of, and this is hard, on whether or not a really cool discovery really has legs for translation. Because like I said, 9 out of 10 or 99 times out of 100, it doesn't. But sometimes it does. So you should certainly flesh out the idea, and you should also talk to people, I would say, that again have done that and have that expertise on what it takes. Because as an academic scientist, you don't know what it actually takes to turn one of your discoveries into down, or to take it down the therapeutic pathway. You just don't. You're not trained in that. Why should you have that expertise? So talking with people that do would sure help. And generally speaking, everybody in the sort of biotech industry, they've all been there in your shoes at one point. So usually people are pretty generous with their time, and they'll sit with you with your data. I do it literally all the time. And then you give advice. And sometimes the advice is, this is fantastic, but for example, I've seen a bunch of things that, the science looks fantastic, but there's no mouse model data or, sort of therapeutic data, like, for example, that we generated with EPO, that was really critical to saying that we could take this, because as I said, modified mRNA might never have gotten out of the tissue culture dish. Mm-hmm. But we showed that we could take it into animals and have a therapeutic endpoint. So I've advised some people to go back and do, animal models and show me that the discovery that you've made can, and they might not have as good luck, that we did, that it worked right off the bat. We could take it into animals and immediately show therapeutic endpoints. They might take 5 years doing that. Okay. And if that's the case, you don't want to start a company, at that point, because you're going to dump a lot of money into 5 years of answering the question, can we take this into living animals? And if the answer is no, that's 5 years of investor money and time that you're probably not going to be able to get.
So in vivo data seems to be a very important thing for you for looking at translation potential.
Yeah, of course.
Okay. Okay.
Yeah. And, there's IP protection, but I think, it's interesting. The CRISPR story tells us it's really, as you know, that you may or may not know, but, the CRISPR IP wars are still on, yet that hasn't stopped multiple different companies coming forward, going public, and going into clinical trials. So I find that really interesting, actually, that the fundamental IP, which there's a battle between UC Berkeley and the Broad, is as yet undecided, and that hasn't stopped these companies from. So I don't know. I think it says something about the importance of fundamental IP. And there's other examples of that too, even with the small interfering RNAs, sort of blocking patents and fundamental patents. Often there's a way to write around, if you make an important discovery in an area and maybe it looks like there's really good IP in that space, there might be a way around it. I don't know. I find that as maybe an evolving sort of area, because it was thought to be really critical. It is really critical, but is it though?
Understood.
CRISPR has told us, is it though?
And in the first few years of a startup company, what do you think are the main challenges at this point that will make or break?
Well, fundraising and runway. I mean, you want to be very careful about, not having only 1 month left and then all your money's gone. So I was recently talking with a company, some great science. They've been at it for, I think at least 5 or more years, and they've got a couple of month runway. And that's a challenge. The team is so important. You want to work with people that have seen the movie before. So if you've seen the movie before, you know what's coming next. If you haven't seen the movie before, you might be like, oh, I wasn't expecting that. So that's important. Track record. And sometimes I think one important fundamental is, when you're first trying to, let's say you've got a platform technology that can be applied to many different things, you don't want to set the technical hurdles, up here when you can set them here. You want to set them as low as possible. Your first application, you want to be as easy as possible to jump over that bar. You don't want to set the highest one. The highest one might have the greatest clinical utility, the biggest patient population, the biggest bang for the buck if you succeed. But your chances of succeeding when you try to jump 10 feet over the bar are a lot less than if you're trying to jump over a 6-inch bar. That's a lot easier. And, as soon as you jump over the 6-inch bar, all of a sudden, that your money comes in because you've shown that you can do it. And all of a sudden your runway changes, and now you can set the 1-foot bar, and you get over the 1-foot bar, and now you can go for the 10-foot bar. So really be careful about what you're setting out as your first clinical sort of targets and applications, that they're not unnecessarily technically challenging.
And in the initiation of a clinical trial, do you think this is something that needs to be built in internally or through collaboration with larger companies, since I assume they must be quite expensive?
Yeah, there are multiple companies that help you design clinical. It depends on the size of your company, whether or not you have the in-house expertise and experience to do it, or you don't. So, but the one thing that's true is that you want to design it properly. You don't want to design a study that then is, you get data that's uninterpretable, or it's not powered correctly, so you can't make a conclusion or whatever it is. So you don't want to drop the ball at that stage. So it sure doesn't help, or it doesn't hurt to get, real experienced clinical design experts looking at your design to say, yes, that's reasonable, but have you thought about this? This might be a better primary or secondary endpoint or whatever it may be, and power it correctly. And so you don't want to screw up at that stage. You brought it as far as getting into patients. You've paid the money to run a clinical trial. Your design better be good.
Awesome. This is very, very insightful advice on clinical translation of basic discoveries. To conclude, I would like to ask you if you have a few key messages for young investigators today. What set of skills would you encourage today's new investigators in hematology or stem cell biology to actively pursue?
Well, I would go back to one of the themes that we talked about earlier, which is don't just sit inside your box, read outside of your box, meaning, outside of your field, to be inspired by other ideas that you might be able to translate into your own work. That's important. Another one that I used to tell my trainees a lot was, well, you might have a really great idea, and it really, if you could succeed, it would be fundamentally game changing. But if you don't have a technological way to address that question, you have one of two options. You can either work on the technology to be able to get past that hurdle, or you can put that project on the shelf and wait till somebody else comes with the way that you can answer that question, then take it off the shelf quickly and answer the question. But you have to know that there's a technically sound way to address the question that you're asking. Mm-hmm. And like I said, we as scientists, we run into walls all the time. That's science. When you're exploring the unknown, you hit a wall, and there's two things to do. You can turn away and say, gosh darn, there's a wall there and it's 10 feet of concrete. How am I going to get through that? Or if you have the persistence and you have the talent and the creativity, you can actually chew your way through that wall. It may take a long time though. So you have to have, runway, and you might need to be really creative to get your way through, and you might need some dental work because your teeth wear down as you're chewing your way through the wall. But I mean, these are the things that this is kind of what I would think about when I was running my lab as a young new investigator. And I would encourage others to, but everybody has their own recipe for how they like to do things.
So creativity, perseverance, collaboration, mentorship.
Yep.
Perfect.
Thank you. You summed it up much better than I.
No. I'm just taking your words, and it's been really amazing to hear your story of knowledge translation and your career path, Dr. Rossi. I'm sure that many people would like to have the opportunity to thank you for your contribution to mRNA therapeutics, given the current context. I will thank you on behalf of everybody who would like to be able to do it. Thank you very much for spending time with us today and sharing your story with ISEH.
Oh yeah, it was my pleasure. Thanks. It was fun. That's well.
Have a nice day.